Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
I want to start today by taking you right onto a manufacturing floor. You can almost picture it, right? The hum of bioreactors, millions of dollars of fluid moving through all that steel. It's a huge, high-stakes environment.
Yeah.
And then in walks a data scientist, or maybe, a very optimistic vendor, and they drop the single most dangerous phrase in modern manufacturing.
I can feel the tension already. They walk in and they say, "We can predict quality."
That's the one. We can predict quality. And look, if you're coming from the tech world, that sounds amazing. That sounds like a win.
It sounds like the future, right?
Exactly. We've got the data, the algorithms. We know what the product looks like before it's even done. But as we saw in the research for this deep dive, that phrase doesn't get applause in pharma. It triggers more like an immune response.
It absolutely does, because the second you say predict quality in a GMP facility, you are not having a data science conversation anymore.
No.
You've just walked completely blindfolded into a regulatory minefield. You've invited quality assurance, the validation group, and the QP, the qualified person, into the room.
And they are not looking at your cool graphs.
Not at all. They're asking one, frankly, terrifying question. Great. If you can predict it, does that mean we're releasing this batch without testing it in the lab?
And just like that, the stakes go from, optimizing a process to patient safety and legal liability.
That's it. And that is the tension we are really digging into today. We have these incredible tools, digital twins, analytics twins, AI models that promise to revolutionize how we make medicines. But there's a massive and I think often ignored gap between a model that works on a laptop and a model that lets you legally put a vial of medicine into a box and ship it to a patient.
So that's the mission for this deep dive then. We're going to explore that gap. We're going to find out if predictive quality is actually reducing the testing burden or if it's just taking the risk from the lab and hiding it inside a model that might be drifting when nobody's looking.
Which is a very distinct possibility.
But before we get to the failure modes, and there are some scary ones, we have to set the scene. Why is the industry even pushing for this? Why take on all this risk?
Because the old way, what we call offline testing, is just becoming operationally unsustainable. We treat it like the gold standard, but in reality, it's post-mortem data.
Post-mortem. That implies it's already too late.
In a manufacturing sense, it is. I mean, think about the logistics. You take a sterile sample, you verify the chain of custody, you walk it down to the QC lab.
Then it sits in a queue.
It sits in a queue for an HPLC, maybe a mass spec, best case scenario. You get data in 3 days.
But the biology inside that tank, it didn't just pause for 3 days while you were waiting.
Precisely. The fermentation is happening right now. The chromatography column is shifting right now. If you're running a continuous process, looking at data from 3 days ago is like driving a car down the highway while looking only in the rearview mirror.
You can see exactly where you crash.
You can see the crash perfectly.
Yeah.
But you have zero ability to prevent it.
So the ambition of this analytics twin, which is the term the sources use, is all about closing that time gap. It's moving from checking what happened to knowing what is happening.
Exactly. And we should be clear on the term. We're not talking about a digital twin of the equipment, like a 3D model of a pump.
Right.
We mean an analytics twin. We're trying to link process states, things we could measure instantly, like pressure, temperature, pH, and use them to mathematically predict the critical quality attributes, the CQAs.
The things you'd normally need that slow, expensive lab to measure.
That's it.
And this leads us right into the first major trap from the research. This disconnect between correlation and decision support. I found this fascinating because it seems like a lot of teams stop at correlation. They get a high R-squared value on a graph and, they think they've won.
That's what I call the science project phase. It's easy to find correlations in historical data. You might find that when the temperature fluctuates in zone 3, your impurity profile shifts. Okay, that's a correlation. But the question that actually matters in manufacturing is, so what? What do you do with that information?
The sources call this the actionability gap.
And it is a huge problem. Let's play this out. Let's say you build a brilliant model that predicts a spike in host cell proteins, HCPs.
Okay.
The model is accurate. It tells you, hey, in 2 hours, your HCPs are going to go out of spec. That's incredible insight. But do you have a knob on the machine that reduces host cell proteins?
Well, if the process is validated and locked down, probably not. You can't just start freestyling with the feed rates unless that's already part of your control strategy.
Exactly. So you've just built a very expensive, very sophisticated fire alarm that rings loudly but doesn't actually call the fire department.
You're just generating data that says, we have a problem, but you're operationally powerless to fix it.
Which, in a GMP audit, isn't an asset. That's a liability. You are effectively documenting your own failure in real time.
Wow, that's a really important distinction. So for a model to be worth the regulatory headache, it has to be tied to a decision you can actually execute.
It has to change something physical. A classic example is chromatography pooling. You're watching the product come off the column, a model is analyzing the UV trace, and it says, the impurity levels are rising, stop collecting now. You execute a cut.
That is decision support.
That's it. You aren't just watching, you're acting.
But as soon as you start acting based on a model, the paperwork changes. The sources put these models into 3 buckets: informational, hold point trigger, and release criterion. I'm guessing the validation burden is not linear.
It's exponential. An informational model is, nice to have. If it breaks, you just go back to the old way. No harm, no foul. But a release criterion model, a model that decides if the product is safe for a human, that is a control element. It is now part of your license to operate. If that code has a bug, you are potentially releasing adulterated product.
And that takes us right to real-time release testing, RTRT. Because that's the dream, isn't it? We can stop doing traditional testing, we can fire the lab.
That is the single biggest misconception in this entire field, and it's a dangerous one.
So, to be clear, does predictive quality mean we can stop testing?
Absolutely not. And we have to distinguish between reducing rework and real-time release.
Okay.
Reducing rework is about saving money. It's using soft sensors to catch a batch that's drifting off course and just nudging it back. That's great. It pays for itself. But that's operational control.
It's like lane assist in your car. It helps keep you on the road.
Perfect analogy.
Yeah.
But real-time release is like getting a license to drive blindfolded.
Oh, okay. So a whole different level of trust.
It's not about trust. It's about proof. RTRT doesn't mean we think it's good. It means we have proven it is good to a distinct level of statistical confidence that replaces the analytical method. And here's the kicker people miss. The attributes still have to appear on the certificate of analysis, the COA.
That's a key point. When a hospital pharmacy gets the drug, they get that piece of paper, the COA, and it lists purity, potency, all of it. Even if you used a model, those lines still need to be filled in.
You aren't removing the obligation to prove the quality exists. You're just changing the source of the evidence.
So instead of a lab result, the evidence is the process data.
But that means the burden of proof shifts entirely onto the model, and the standard for that proof is defensible in a court of law.
This has to be the 95% problem from the reading. In operations, if my soft sensor is 95% accurate, I'm thrilled. That's great.
You're running the plant better than yesterday? But for release, 95% is disaster. Imagine telling an FDA auditor, we are 95% sure this batch is sterile.
Yeah, you'd be shut down before lunch.
Right. If you're replacing the release test, you need to show the model covers the whole design space, and this is critical, that it doesn't suffer from the tails problem.
I wanted to dig into that, the tails of the distribution. The notes flagged this as a technical detail that kills a lot of these projects when they try to scale up.
It's a detail that seems small until it completely ruins you. When we see 98% purity on a spreadsheet, we think of it as a single data point. But biologically, that purity is an average of the entire batch. In reality, in a big tank, quality is a distribution.
So you mean physically inside the tank, or just statistically?
Both, really. You have isoform distributions, impurity profiles. Most of the batch might be super pure, but there's a tail of the distribution where the impurities are concentrated.
And predictive models. They love averages.
They're obsessed with averages. They're mathematically designed to find the line of best fit. So an analytics twin might predict the average quality perfectly. It says, overall, this batch is fine, but it might completely miss the fact that 2% of the batch, the tail, is toxic.
So you release the batch based on the prediction because the average looked good, but you've actually sent out dangerous product because the model just smoothed over the risk.
Correct. If you don't explicitly train your model to look for the tails, to look for the extremes, you are blinding yourself. You're trading safety for mathematical simplicity.
That is a terrifying hidden failure mode. But there's a more obvious one that the sources spend a lot of time on, the concept of lifecycle management.
If there's one thing I want listeners to take away from this conversation, it's this. Building the model is the easy part. Keeping the model alive and validated is the hard part.
The text calls this the day 2 problem. You launch the model, everyone celebrates, the data scientists move on, and then drift happens.
Drift is inevitable. It's just physics. Sensors age. A pH probe today does not read exactly the same as it did 6 months ago. Your raw materials change. The biology shifts.
And the model, which was trained on that old world, starts making bad predictions.
Right. And this is where model lifecycle management becomes a core GXP requirement. It's not enough to say it works.
Yeah.
You have to have a system that detects when it stops working. You need drift detection triggers.
There was a specific nightmare scenario in the notes about ad hoc updates. It sounded like the way software companies operate, pushing patches. Why is that so bad here?
Oh, the Tuesday patch.
Oh.
This is where a data scientist notices the model's drifting. They think, I can fix this. They retrain it on the last 5 batches, improve the accuracy, and push the update. In Silicon Valley, that's continuous improvement.
But in pharma?
In pharma, that's a violation of federal regulations. You've just changed the yardstick. Imagine you're measuring a table with a ruler. Then someone swaps your ruler for a slightly different one. You can't compare the measurements anymore.
And looking back, you can't audit it.
That is the real danger. An auditor comes in 2 years later and asks, why did you release batch X? You need to be able to reproduce the decision. If you overwrote the model version that was used for batch X, you can't. You've lost data integrity. You have to treat the model code just like a physical test method.
You wouldn't let a lab analyst just tweak the HPLC method on a Tuesday because they felt like it?
Never. This links right back to the FDA's process validation guidance, stage 3, continued process verification.
Right.
The FDA expects you to know if your process is drifting. If your monitoring system is the thing that's drifting, you're flying blind. You need a meta-monitor, a system that watches the model.
It seems like this creates a huge organizational void. Who owns this meta-monitor? Who owns the life cycle? The sources suggest it's a bit of a hot potato.
It is the classic silo problem. Quality assurance knows the regulations, but they don't know Python. The data science team knows the math, but they don't know GMP or change control. MSA understands the biology, but maybe not the software infrastructure.
So when the model drifts, everyone just sort of looks at everyone else.
And the model guys. I've seen it happen a dozen times. A brilliant pilot project works for 3 months, then the inputs shift, the accuracy drops to 80%, nobody knows whose job it is to retrain it, so the operators just turn it off.
Turn it off, the death knell of digital transformation.
It is, and surprisingly, that's usually because of what we call the input problem, which is another scaling trap. When you try to move these models from site A to site B tech transfer, they almost always break.
Even if the process is identical.
Well, the process is never identical.
Yeah.
But usually it's not the biology that breaks the model, it's the infrastructure.
Yeah.
Maybe site A samples the reactor every 4 hours, and site B samples every 6. Maybe site A uses Mettler-Toledo probes, site B uses Hamilton probes.
To a human, those are small details. To a machine learning model.
To a model, those are completely different worlds. The model is learning the noise of the sensors just as much as the signal of the biology. Change the sensor noise, the prediction fails. That's the input problem.
So the whole idea of fleet learning, where all your factories learn from each other in a giant cloud brain, is a bit of a myth.
It's a great marketing slide.
Yeah.
In practice, it is incredibly difficult. Unless your factories are carbon copies down to the brand of the thermocouples, it's very, very messy.
Okay, so we've covered the risks. The actionability gap, the 95% trap, the tails of the distribution, the life cycle nightmare, and the input problem. It feels heavy. But the industry is moving this way, so how do we do it right? The sources gave a framework for governance.
It starts with a mindset shift. You have to stop treating these as IT projects and start treating them as engineering control projects.
Okay, so let's role play a design review. I'm the facility manager. I come to you with a shiny new AI tool that predicts product purity. What's the first hard question you ask me?
My first question is, are you predicting the critical quality attribute directly, or are you predicting a proxy?
Let's say it's a proxy. I can't measure purity in line, so I'm measuring, let's say, turbidity and refractive index and correlating that to purity.
Okay, then I'm going to make your life difficult. Because proxies require justification, you have to prove to me that the correlation holds up not just when things are going well, but when they're going wrong.
Mm-hmm.
If the process deviates, does the proxy still track the purity, or do they decouple?
That sounds like stress testing.
It is. And that's the second takeaway. Build a model stress protocol. During your PPQ, your process performance qualification, you need to actively try to break the model.
Don't just show the good data.
No. Show me data where you deliberately fed it bad inputs. Inject noise. Simulate a sensor failure. Simulate a feed disruption. I need to know where the cliff edge is. If the model fails, does it fail safe? Does it scream, I don't know, or does it confidently predict quality is good?
That distinction is vital. A model that admits ignorance is safe. A model that lies is dangerous.
Exactly. And that leads to the really practical governance step, the drift window.
This is the what-if scenario the text mentioned.
Right. Let's say your model drifts on a Monday. You don't catch it until Friday. You have produced 5 batches in that window. The model said they were fine. We now know the model was unreliable. What do you do with those 5 batches?
I assume you have to quarantine them.
You have to quarantine them, investigate them, and probably retest them with the reference lab method. If you don't have a plan for that drift window, if you don't have the budget or the staffing to handle that investigation, you are not ready to turn the system on.
That is a very sobering reality check. It sounds like the technology is actually the easy part. The governance, the people systems, the fallback plans, that's the really hard work.
That is almost always true in manufacturing. We get enamored with the algorithm, but the safety comes from the protocol that surrounds it.
So, to summarize our roadmap today, predictive quality is the future. It's better, it's faster, and eventually it will be safer than offline testing. But it shifts the burden of evidence.
It shifts the burden from the lab to the process data. It does not remove the obligation to demonstrate control. You're still on the hook.
And that leaves us to the final thought to leave with everyone. The sign-off.
This is something I tell every client. Next time you are in a meeting about digital twins, look around the room. Ignore the code, ignore the dashboard. Ask one single question. Who signs off?
And not just who approves the budget.
No. Who puts their signature on the line to say, this model is in control? Who is the qualified person equivalent for the algorithm?
Because right now, in many companies, that person doesn't exist.
Correct. And until that person exists, until someone with authority stands up and accepts the risk of the model's performance, you don't have a GMP system. You just have a very expensive graph.
Who signs off? A simple question that cuts right through all the hype.
It's the only question that matters in the end.
We'll leave it there. For everyone listening, check your sensors, watch your drift, and we'll catch you on the next deep dive.
Stay in control, everyone.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.